Yeah, so just to repeat again for the sake of recording, it's great to be here. I think that UKRET is a brilliant initiative, and it aligns spot on really with what it is that I'm interested in doing at Genomics England. So I do know a fair few of you, so it's great to see you anyway. And for those of you who don't know me, it's a great pleasure to be in your company. I'll just give you a little bit of background to who I am. So I'm Charles Stewart. I'm head of patient and participant engagement at Genomics England. I'm a genome scientist. I've worked on the human genome since 1994. Yes, 30 years next year. Amazing, isn't it? And so I spent my entire career working on the human genome. But I think probably of more interest to you, and for the sake of today's conversation, is that I have two children with severe neurological disorders, one of whom had West syndrome at 8 months of age. And it was sort of that overnight thing, which I'm sure I don't need me to tell you, but that pivot from that normality to suddenly the whole world of advocacy opening up in front of you. So I have a dad hat, or a dadvocate, which some of you, it's not my word. I wish it was. It's clever. Also a patient advocate. So I interact with a lot of patient groups, and a genome scientist. So it's probably a slightly interesting mix of things. So first of all, I'm here today to represent Hope Epilepsy, which I'm sure a lot of you know. I'm a patron and trustee for for this little charity, and we provide a sort of a safe environment for families to meet up from time to time on Sundays during the year, and we also donate anti-suffocation pillows to people, and I think we also have some seizure alarms, but if you want to don't ask me. I'm not sure how many we have, but anyway. So Hope Epilepsy is a patient group, family group, but we're beginning to sort of spread the tentacles a bit now and interact with more groups. And secondly, I'm here as part of CRELD1. I don't know if any of you have had a chance to look at the website, but CRELD1 recently discovered, and the site was set up by Adam Clatworthy, who had a daughter, Lola, who sadly succumbed to it a year or two ago, and I think it was just before she died, they'd had a little boy, and he sadly also has the same epilepsy. So it's a new epilepsy. It's a great website, and I think it's a really nice example of the power of what patients can do. I don't need to preach this to you, but it's yet another example of, patient taking control, bringing together researchers, clinicians, putting out as much information as we know possible about that disorder, and really getting a conversation going. There's already a paper in peer review for this. It's only been known for a year or so. And we've got some really good members, interesting members on our scientific advisory board, including Nick Lench, who is the executive director of NATA, or Pete's boss, I think, who will be speaking later. So anyway, as I said, I'm head of patient participant engagement at Genomics England. I've been there for 3 months, so I'm still learning what I can and can't say. Although, to be fair, I've been involved with Genomics England a fair amount longer than that, because my daughter's on the 100,000 Genomes project. So I've been part of the participant panel since 2016, although I resigned my position when I joined Genomics England. And I have a particular interest in epilepsy. Obviously, that's why I'm here, but also a particular interest in epilepsy at Genomics England. And one of the things I'm tasked with is to see how I can bring about more value. I'm sorry, it's a horrible word, isn't it? But it doesn't imply anything to do financially, bring more value to the very large database that we have at Genomics England that contains all of our genomes. So I don't even know how many there are in there at the moment, but the tens and tens of thousands of them. And there's a very significant number of people with neurological disorders and, of course, epilepsy. What we're really interested in doing is bringing together as many researchers and clinicians as we can to use these data. We do have a few people who are doing it already, but let's bring as many people as we can into it. Globally, we want to bring more people into. But more importantly, we want to bring in the patient voice. So we're looking to work with disease-specific charities, so obviously epilepsy charities, and epilepsy patient support groups to see how we can help them in their journey, how we can perhaps introduce them to various different people and various different fields of research, perhaps, that they're not aware of. Generally, I suppose you can think of me as a matchmaker or the midwife at a birth. So I'm just there to sort of facilitate getting the right people talking to each other. And what I would like to know is, open floor, what are the major, or what are the minor challenges that you've all had, getting your groups up and going? Who, have you been able to meet researchers? Do you have enough patients? Is your disease so rare that you've only got a few other people? Could it help you if you were to have access to the research environment, as we call it? So the research environment, that's where the data is available from. What other problems have you got? And is there anything that you think I should be able to take back to Genomics England that might help you? I appreciate, that question could really spiral out of control. We can talk about everything to do with genomics and everything to do with sort of therapy, etc. But sort of practical measures that you think I might be able to help with first off. And we're looking to have just to follow that up, we're looking to sort of champion epilepsy this year, and we'll be hosting webinars, I hope, with specific researchers who are working in Genomics England or working with Genomics England. But also it's really important that we bring in the patient voice as well. And we've got a sort of an exemplar for that already. We've run 2 webinars so far for sight loss, so the genetics behind people who have sight loss. And it's if you get the chance, I appreciate it's not related to epilepsy, but it is, we think, is quite a nice way of, instead of just sort of sitting there in front of the slides, people going through all this sort of technical stuff, we're actually having more of a conversation, which is being led by our head of public engagement, a lady called Viv Parry, who you may have heard of. She's a journalist. She used to present Tomorrow's World, and she's really good at that sort of thing. And it's a way to get people talking rather than just sitting and listening to or watching slides. So getting a couple of experts in there, perhaps getting a couple of charity representatives from epilepsy, and then everybody else in the audience being able to ask questions. Right, so that's me. Who wants to go first to answer my questions that I put in the email? Or is everything fine? I really, really want to see what I can do to help.
From Cath Research, which is a really new charity, so I don't expect anybody here really to have heard of it.
Could you put your link into the chat just while?
Yeah. There we go. And the difficulty we found is trying to, one of your first questions was trying to, I've literally been spending so much time just scouring the internet, trying to find universities that work in neurological disorders, trying to find UK scientists that work in epilepsy. And it's just extremely time consuming. The people I have found have responded. So we've all had like kind of positive response. Well, not positive responses, but, you know, they've actually responded, which has been very nice. But it's just been very, very difficult to try and find the right people to reach out to.
Yeah, that makes sense. That's a clear and obvious problem. In my experience, from what I hear from other people, sometimes finding clinicians isn't such a hard thing to do, but research can be much tougher. Bearing in mind, clinicians are trained how to deal with people, how to cope with difficult situations in passing. I don't need to tell you this, of course, but research is very different. Researchers may often work on genes, which are really fascinating genes in nature, and many of them may not have an understanding of the sort of the practical challenges of what it is like to when this gene goes wrong. So there are sort of do's and don'ts of how you approach a researcher, and I certainly know people in the past who have gone to them saying, you might be able to save my child's life, and of course that sort of thing makes people run for the hills. So there's a way of sort of tempering things, but no, that's just a comment. That's not necessarily anything to take on. So I'm very sorry, I don't know anything about CASC. Is that the name of the gene?
Yes, yeah, the gene is called CASC. It's mainly obviously in the brain. So 40% of our children have epilepsy, mainly infantile spasms, but it causes severe learning disabilities in everybody. And I have obviously found some scientists online, but they're already involved quite deeply in other diseases, and they've all replied that they don't have like the space within their organizations to take on a new disease. We found one, we've linked with Bristol University, and they're hopefully going to do a bit of work, but obviously the more the merrier. And I didn't know if there's like a secret list somewhere.
Yeah, so how many people do you know who have this in the UK?
I personally know about 23.
And do you know globally?
Yeah, around 230, but that's literally the people that have put themselves out there and have found social media. Obviously, there's no kind of definitive number, but we have 230 people kind of registered on a very general like subscription.
And do you know how many of those people would have been diagnosed through the 100,000 genomes or the Genomic Medicine Service?
I don't know the number, but I do know quite a few in the UK have been, because it's you can get quite a spectrum, and the much milder ones, I think, were only diagnosed by that because the general condition shows a condition called pontocerebellar hyperplasia in the brain, which is quite clear on an MRI. So they're generally, and they all have microcephaly. So I think the diagnosis is fairly good, to be honest, unless you're on like the very unusual mild spectrum, where it might be like a mosaic mutation or like a kind of mild missense or something. So, and then they have been discovered on the 100,000.
Yeah, and how long has this been known for?
The disorder was discovered in 2008.
Okay.
Yeah.
So that was when the first time that the gene was found to be causative.
Yeah, so the gene's been looked at for longer than that. But
and do you know who was the do you know who the publishers were of that paper? Have you contacted?
Yeah, she's actually on our scientific advisory board. It's a German researcher called Kirsten Kutsch. So she's just joined our team.
But you would like someone from the UK as well?
Yeah, we need so literally we have one definitive person on the scientific advisory board. We have a neurologist who's helping us, but he hasn't committed to being on our board because he's already on 2 boards, CDKL5 UK and another one. And yeah, I think it'd be nice to have another person and just some other links maybe to other institutes that might be interested in kind of neurology, epilepsy, rare disease, or even kind of biotechs, or anybody that might be interested in the gene that has space for us.
Yeah, I'm really pleased to hear you say you've got a scientific advisory board, be it one member at the moment, but I think that gives you a lot more credence it shows that, you know, well, it shows your ambition and your intention. Because yes, I've certainly, you know, some groups seem to sort of have a, I don't know, if they don't have a sort of a research purpose, they can sort of dwindle after a little while. I think after the initial enthusiasm disappears. But that's just my opinion. Of course, I could be completely wrong. But that's really helpful. Thank you. I'll have a look at your website in more detail after the call.
Oh, thank you.
Right, Tony.
Laura, sorry, Laura. While you were chatting, because I found myself on your website the other day, I went on to Mastermind, and you've got 9.5 thousand papers that have been written about your gene. So each one of those will have authors based in different places. It's going to take a while, but if you look at all the authors, you'll find out where they are.
Yeah, thank you. I have obviously done that, but I think part of the problem is there are quite, I mean, we're lucky there's quite a lot, but it's difficult for, yeah, it's time consuming. So that's how we found Kirsten, and I kind of knew about her anyway. And yeah, but sometimes it's easier if there's fewer papers in that respect.
Charlie, I had a question. It was raised by a patient, one of my members, when asking of a clinician who works in one of the centres of excellence that we luckily have in Behçet's, unlike many rare diseases, would they be genome sequencing Behçet's patients? And the response came, no, they're only interested in monogenic diseases at present. And I suppose, well, my question is that strictly speaking true? The difficulty seems to be it is such a complex area. One doesn't know where, you know, if you don't know where to start looking, it's a complete nightmare. But whether or not things like using sort of different tools and techniques, I mean, doing long read sequences is an example for searching for things where you stand more chance of finding stuff, whether when that comes in and is used more would assist. But you see, Behçet's has a problem. They back in 1990, some Japanese chap doing some research found a gene that they thought was associated with Behçet's. It's called HLA-B star 51, and the trouble is only 30 to 40% of people have it, and they prove associations by regression analysis. And if I was cynical, I would say that actually they thought it was a silver bullet, and it's been peer reviewed so many times, it's very difficult for people to back out of that thing. Because ultimately, as I say, 34 don't know. It's more associated with eye disease. You can go blind very easily, which is why the Japanese are very interested in the whole thing. But it's sort of, you know, when you get a problem like with Behçet's, 20% perhaps of our patient population have neurological problems, of which some of those, but not all, have seizures and stuff like that. Don't know whether you call that epilepsy. I'm not, you know, technically qualified for that. But it's a real conundrum because it affects any organ in the body and all the rest of the stuff. So I suppose that, and the impression I got sort of, you know, the 100,000 program was effectively, they quite rightly, it was a good way to start the program, targeted things that they sort of knew would have a success. And then all of a sudden, doing all the research, they suddenly found other issues that came up, one of those blood clotting things that came up very early in the program. And I just wonder whether we're actually adventurous enough, because I'm the Alliance Network Chair of Central and South Genomic Medicine Service Alliance, and I'm very interested in getting at the populations that historically we haven't. And the problem is there's no point me firing up everyone in Birmingham to get involved in the genome area if they're just going to be restricted on what can be tested because of what's on the National Genome Tester. So really, not just have a PPI committee, people into communities and sound them out and talk with them extensively, not just a one-off sort of research PPI exercise, but an enduring thing that will last when I'm dead and gone. I've said, but I mean, the fun speaks to to best advantage, or are we just you know, you look, if we're not careful, we're going to miss out on the ethnic communities, and we're just going to reinforce what we already know.
So yeah, some really interesting, great points there. So the newborn sequencing program, the disorders they're looking at there are specifically those that have a treatment for them. Admittedly, I hope very much that that number will increase very much over time, assuming that this the pilot study proves to be really useful. The diverse data again is that sort of the ethnic makeup of the different people going through genomic sequencing. That is a big deal, and there is an initiative at Genomics England called Diverse Data. I don't know if you've heard of that, but when it comes to so are you saying that there isn't the sort of the pathway for people to with Behçet syndrome to actually be sequenced through the NHS? Well,
it alludes to a wider problem that lots of rare diseases have. Everyone needs, if you look at the conditions that start having success, they have a patient registry, then they do a natural history study, then you really get it, you understand your populations. And a lot of the small charities, and I'm trying to sort that out for my society, and I'm finding that difficult enough. Research excellence framework where people are doing papers, getting cited, doing reads, and this, that, and the other. There's no research. I've been, I said 5 years ago in one of my newsletters, which I know all the clinicians see, I want what I call a technology roadmap or a research roadmap, so we all know where we're heading collegiately together. And sort of, one of the things that happened during COVID allegedly was, well, there was a more collegiate approach across research. And you sort of indicated when you're chatting to the previous question about, if you find a useful researcher that's interested in your topic, well, how do you do that? Well, one way is doing a James Lind Alliance. So you get your top 10 questions. But I try to get my trustees to agree with that because it costs about 80,000 quid and they weren't interested, but all of us need that. And my concern to clinicians, the vast majority just continue as we are, frustrated, and we talk about equity, but I'm not too sure it's in any guise.
Do you have a website you can share?
Yeah, I'll do that. Anyway, I spoke for long enough.
No, thank you.
I think there's a massive great issue to do with how research is conducted, for whom, and I still think the rare diseases in general, not just mine, I'm not fussed about mine, I'm sorting that out, at a real disadvantage. And that's why I think Alison's initiative with RET is really important, because there's so many. And I've now got actual patients, it's even worse, who a clinician in one of the centres, a neurologist now left, and the patients are being told they can't have a plasma because it's not and the patients are being told they can't have, so they're now having to find some champion neurologist somewhere who's going to fund their treatment. I mean, it's just, you couldn't make it up. It's just disgraceful.
To more today, but I'm very interested.
They can't have a plasma pheresis.
I have my email today, but I'm having a look at this thought so far.
But because it's, I mean, it's just, you couldn't make it up. It's just disgraceful.
Alison, shall I make a start? Some few points?
Yeah, absolutely. Go ahead. Thanks, Peter.
Yeah, great. Really nice to see everybody. A couple of familiar faces. Jonathan and I met through some SYNCAT work, and Charlie and I met last week to talk about the old days of working in the lab with the old days of the old pre-genome era, as they call it. That's great. So yeah, thanks. For really good to see you. So just to explain really briefly. So my background is in I guess basic lab-based research. I've been a researcher in the University of Oxford and a program leader at the Medical Research Council. I'm now head of biology at NATA, So NATA stands for Accelerator, or NADA, if you're from America, as we found out in our collaborations. And really, we're here to kind of improve the field of, particularly in rare disease, of novel therapeutics. So this is a really exciting new area of therapeutics, and some of you may know this fairly well, but it's very, very new. It's just the first examples are just in the clinic now, so this is super new. So it's very, very early days. But we're really here to understand more about patient population, where there's opportunities to collaborate and find new therapeutic opportunities, and also to understand some of the basic biology behind particularly seizure-related disorders. So I have a personal interest in epilepsy through my previous research. I worked on pediatric epilepsy models in cells and in other systems. And as Charlie mentioned, our executive director here at NATA is Nick Lench, and he's on the board at ERUK and has a long-term interest in, obviously large-scale clinical genetics, but also rare disease and rare epilepsies. So between us, we have a nice, mixture of interest, academic and social interest, and public patient integration in epilepsies. So I'm just going to share a couple of slides. There's a little bit of science in here, but I'll go through it very quickly. Just want to give you a sort of taste of what we do, and there's something coming up with some new funding, which I hope you'll find interesting around opportunities for rare epilepsies, and I'm involved in that project. So I was going to share my screen briefly. See that? Okay, great. I won't go through too much, just be very brief. So just tell you briefly who we are, where we are, what we do, and then discuss a few points that might raise some discussion points about future opportunities in epilepsy. Just to reiterate, this is really new research, really exciting, really cutting edge stuff, and it's very early days, but I think it's a really good chance to engage with people now and think about opportunities for therapeutics in the future. So just to reiterate that. So we're based on the Harwell campus. That's about 10 miles south of Oxford. It's just south of the pretty town of Didcot. And we're on a very exciting scientific campus here. There's physics, engineering, space science, lots of biotechnology, etc. here. So we're on a really sort of vibrant scientific campus. We're funded by what's called the Strategic Priorities Fund. This comes straight from the government a few years ago to really say they wanted to push this type of technologies. So they're giving us the money to really push this area very specifically. And our really aim is to kind of understand more about these therapeutic options, understand the biology, and also work with pharmaceutical companies to try and get some of these things into the clinic over the next sort of 5-10 years or so. We're not focusing just on epilepsy, just to be clear. We have as I mentioned, we have a personal interest and a really important interest in these types of disorders. We know there's a real clinical need, particularly in rare epilepsies, pediatric epilepsies as well. But we do work on other modalities as well. We're also working in other disorders outside of the nervous system, just to say. So just briefly, here we are, just briefly. So, sorry. So Nick Lench, some of you may know, he's, so that's the animation there. He's our executive director, and he's come from many years of running genomic services. He ran the genomic service at Great Ormond Street Hospital for many years. And as some of you may know, he's also involved in a commercial genomics company and sequencing company called Genetica, which Charlie will know. So really strong pedigree in clinical genetics, large population genetics, and understanding the role of rare disease. As I mentioned myself, as head of biology, so I've been here since last year. And we're really excited to have Mark Cunningham with us, and he's come with 25-30 years of experience in big pharmaceutical companies. So he worked at Janssen, so that's Johnson & Johnson. And so we have that real experience of people understand about pharmaceutical companies as well as an academic interest. We have a nice mixture of classical genetics, sort of lab-based bench science, and also sort of operations, understanding how this type of thing works. As mentioned, we're on this Harwell campus just south of Oxford. I won't go through this now, but there's lots of really fantastic science here. Some of you may recognize what we call the donut, which is this thing down the bottom here, bottom left, which is it's for doing a sort of hardcore, I guess protein structure and structural biology and physics. But that's one thing you may recognize. You may see it if you drive down the A34. So just really super brief to describe the biology behind the things we're looking at. So this is basically these new tools for genetic disease therapeutics. So some of you know, obviously, you have your DNA, and some people may know some of these disorders are caused by changes in the DNA, maybe one small change in your DNA. So what we can do in these systems is actually sort of find a way to either control or dial down or dial up the particular piece of DNA and the RNA that it expresses. So how it works here, I was going to get my point quickly. For these short pieces of nucleic acid, there could be a mixture of DNA and RNA. These are small pieces. Some of you, obviously, most of you, I'm sure, have had your COVID vaccine, and that's actually a similar technology. This is the same idea, but much smaller piece of DNA and RNA. And how it works is it actually can find its partner, this little piece of what we call an antisense oligo, can find its partner RNA, this RNA sequence in the cell, and it can actually cause that piece of RNA to be degraded and stop the protein being synthesized. Okay, so the idea being, if you have a 2 copies of a gene and 1 copy is defective and producing a protein which is causing problems in the cells, then you can actually use these drugs, these little bits of nucleic acid, to actually target that RNA and stop the protein being produced. So just to be clear, this is a bit more like a normal drug. This isn't like CRISPR. So CRISPR, as some of you may know, that's a permanent one-off change in your DNA. That's something very, very different. This is much more of a temporal change, a sort of a short-term change in expression of a gene. So that's very useful. Therefore, you can obviously switch things on and off. You can take a dose and then stop taking it, etc. So it's much more accurate in terms of dosing of protein of interest. So that's what's called removing toxic protein or dialing down expression as a gain of function mutation. Also, we can do the opposite and actually switch something on or turn something up, dial something up. And then a similar system, slightly different chemistry, but same idea. This time we actually can take a short piece of DNA and RNA. It's a slightly different type of structure. This then goes into the nucleus of the cell and then binds to next door to the gene of interest, attracts the systems in the cell to switch on the gene, and then produces more of the gene and more of the protein in the cell. And that's very useful if you have, again, 2 copies of the gene. One is defective, but the wild type one, the other copy is working properly, then you can amplify and make more of the wild type, the normal copy, and that'll take over the function of the one that appears to be defective. Okay, so you have what's called gain of function or loss of function, different types of modalities of types of mutations you may see in disease. You can actually address both of those in different ways. Okay, so give you an idea. So it's just 2 different ways of looking at the types of mutations that are involved in rare disease. So it's very, very flexible for that case. So we've got funding to work here in the NATA hub. So that's where we're here based in Harwell. So we have a chemistry department and a biology department. We've also funded some what's called challenges, which I won't go through today, which is where we funded some really large collaborations to work on some aspects of the technology behind these ASOs and these types of antisense oligos, etc. That's a mixture of academic partners and also very, very large pharma. Some of the big companies, AstraZeneca, Ionis, Wave, some of you may know the names. So we are working with some of the top people in the world on these technologies. So we're really working with the best people in this area. So we've got one working on manufacture of these bits of DNA and RNA. That's the whole aspect which we won't go into today. So obviously you have to make these things, to make them clean enough and good enough quality to be put into patients. And the other one's a very large consortium headed up by the University of Oxford, as you can see, with lots of the big players in academic and also pharmaceutical industry to try and how do we improve delivering these types of modalities in patients, which is obviously a key feature. So we have a chemistry lab, as I mentioned. Here's some pictures of our chemistry lab. That's Andres, one of our chemists, and that's Xiao, one of our biologists. So we can make these bits of DNA and RNA, these oligonucleotides as they're called. We can make them ourselves. We can do special chemistry to change the way they function in cells. And we can add things to them so that they target the right cells of interest. So we can do lots of clever chemistry to try and make sure these things do the job they're supposed to do. And I'm head of biology, and really our biology capabilities are really to understand basic function of these oligos, these ASOs and cells, testing them in cellular systems, see if they're working properly, changing the chemistry to see if we can improve their function, and obviously testing them in cells to make sure they're targeting the right genes. So lots of basic biology to do to understand how these things work. So as I said before, this is very new technology and there's lots of basic biology to understand. So basically we have this sort of pipeline, if you like. Sorry, this is a bit of a busy slide, but just you get the idea that we basically have a pipeline all the way from somebody coming to us saying, potentially a clinician might come to us and say, we have a cohort of patients and we like to target this particular gene. Can you help us design some reagents to see if we can potentially test this in some cells, perhaps first of all, and then obviously into maybe some other systems later, all the way through to testing things all the way up to in vivo systems such as the mouse. So we go all the way through from research design all the way through to trying to see if we can generate things that work in a full biological system. So just moving on to epilepsy, and of course, this, as I mentioned, this is also the focus of today. So of course, there's lots, I'm sure you're all very aware of, there's lots of different targets in epilepsy. Some are very, very well characterized. And as Tony mentioned previously, some things are much more complex with what's called comorbidities or other features of a clinical presentation that come on different times of life that may overlap in different patients and may vary between families. That's very important. As I mentioned, we can look at if there's gain of function, so if there's a new sort of toxic function, if you like, or unusual function of a gene compared to loss of function if one of the genes is not working properly. So there's ways we can look at both of those. And of course, there's lots of general pathways involved in epilepsy. So how the seizures are switched on, how they spread in the brain, etc. There are some channels that control the way ions move in brain cells, which can be targeted. So lots of ways to think of novel ways of thinking about therapeutics in epilepsy. And also, of course, if you think about, as I'm sure we discussed about clinical outcomes and endpoints, of course, as they're known, how do you assess these things later on? Also, in several years' time, if you get to a clinic, of course, there's lots of things you can do in epilepsy. Obviously, you can use EEG, you can use other ways to measure their outcomes. So I think this is some great exciting opportunities for the future, because there's lots of things, lots of places to start, and also there's lots of ways of measuring if it's successful or not in the patients. So really keen to work with with groups, particularly patient groups, and sort of get really fact find and find where there's new opportunities for these types of therapeutics in the future. Like I said, it's very, very early days. There have been some very early testing in patients, but it's really an exciting time of working out how well these work and whether we can work with groups to identify some new targets. I'm really keen to here to facilitate and then also accelerate, and also includes in the name, accelerate current research in this area, and also work with people that have something that's established, but also to find new areas or new genes or new targets that might be amenable for therapeutics in the future. So that's we're really here to help people and not just to copy what's going on already. In terms of this system actually working, well, there's been some proof of concept in mouse models, and I'm sure most of you are aware there's many of these genetic diseases have a model in the mouse where they've actually made the same genetic change in the mouse to cause seizures or epilepsy. We can argue a lot about how useful that is, of course, but as a tool to see if these things are working in a whole body system in the brain, there's been some successes. And there's several examples here of both upregulation of genes, and some of these genes some of you may recognize, I'm sure, and also ways to switch off genes or turn down their expression. So again, there's definitely evidence that these types of modalities will work. There's lots to do to get them into the clinic. Some of you may know there has been some very early clinical trials of these in sort of seizure-related disorders. This was an N-of-1 trial, some of you may know this very well, from Batten disease, run by a very big team of people, and Tim Yu, again, some of you may recognize his name. So sadly, the patient died a few years later after treatment, but there was some clues that these modalities were working, seizures were reducing slightly in the early stages of this type of modality. So I mentioned again, these are very early days, these are very early trials, but hopefully these early trials are paving the way for much more routine options for these types of things, and obviously cheaper ways of producing these types of drugs in the future. So just finally to say what we're involved in at the moment is there's some new funding being announced by the MRC, the Medical Research Council, it's a UK-wide organisation, and they're focusing on funding some new initiatives in rare disease. So I'm heading up what's called a node. So the model is called a node. So basically, there's going to be 10 or 12 of these rare disease groups, if you like, around the UK. They're going to be focusing on all sorts of different diseases. Some will be very much genomics led, so lots of sequencing, long read sequencing, data sharing of sequencing, that kind of thing. And some will also be focused around a particular disorder. Okay, so we're focusing on pediatric epilepsies and seizure disorders, where we want to find new ways of testing some of these new drugs and new types of technologies, such as the ASOs I've just shown you. And what we're going to be doing is working with some of the best people in the country that work with stem cell models. So the advantage here, of course, is that, as you may know, you can start to model some of the features around seizures and epilepsies in a dish, in a cell culture system. And this is particularly important in rare disease when you may have one particular mutation or genetic change, and the only model there is the patient themselves. You can't necessarily find it in a mouse or a rat, etc. You have to use the patient themselves. As you know, perhaps you know, you can take cells from a patient if permission has been given, and you can convert those cells into cells a little bit like brain cells. And then you can carry out some really detailed physiology, so understanding the electrical firing of the cells. You can make what's called, these are called assembloids. So these are basically groups of cells that have been derived originally from the patient cells, and they've been turned into neuronal like structures. And actually now you can carry out really detailed physiology analysis of these. You can actually carry out what is called multi-electrode array. So lots of electrodes being put on these sort of 3D structures, a little bit like mini brains, and they can tell you about the features that might relate to therapy and firing of the brain. So it's really exciting times and really exciting ways to model these types of disorders. And this is really the future, I'm sure, of screening for new therapeutics will be with this type of technology. It's very new. It does work. We're going to be working with some of the best people in the country on this at UCL and Cambridge and elsewhere in Edinburgh who have these things set up. And we're going to be setting up a new platform to try and improve and really standardize the way we can actually start to screen new therapeutics using these types of stem cell models. It'll be starting just with some wild types, so normal patient cells first, and the idea is if it goes well, we'll then expand into testing some more different type of patient cells to see if we can find some patterns. So really early days. So this is the rare disease platform. So we're going to be working with the Oxford Drug Discovery Institute, which is based just down the road, and they have all the really high throughput technology to look at lots of these little human mini brains, if you like, in sort of hundreds of wells at a time. So we can start looking at screening some of our oligos or ASOs. We're looking at some of the best people in the country working with physiology. So Gabriele Giannani, who's got fantastic work in gene therapy in epilepsies, and also with the Cambridge Stem Cell Institute, and they have some really fantastic tools for making these mini brain-like structures. We're going to work with the University of Edinburgh in data analysis and data integration. This is not quite my area, but of course, you imagine generating lots of data from these types of experiments, really complex. And what we want to do is compare that data from our cell lines back into maybe what we might get from a patient, such as EEG. So can we actually start to predict what our therapeutics might be doing based on what we're seeing in the patient's EEG? So that's some way off, but that's the future of trying to understand how do we integrate the data sets between these different modalities. And also what we're really keen to do, obviously why I'm here today, is to keep the momentum going with rare epilepsy charities and patient groups. As discussed, I think it's a really important time to bring the patient groups together to find where there's clinical needs, obviously support network, but also where there's a biological interest there to find new targets, find new ways to model disease, perhaps do some early biology, and sort of see if we can start to introduce that biology into our pipeline we'll be building over the next 3 or 4 years. So just remains to be, and the last thing to mention is that we're also funding a fellowship with ERUK. This is a 2-year postdoctoral post. The applicants are being interviewed in a few weeks' time. So it shows us again show we're really committed to thinking about the future of research in epilepsy and funding people hands to do experimental epilepsy and improve the future for therapeutics in the future. So yeah, we're excited about that, and that'll be announced fairly soon. So that's the future for NATA and epilepsy. So thanks very much. So just a little whistle-stop tour of what we're doing, but I hope that's given you an idea of something really exciting and new for the whole field. It's very early days, but there's lots of really fantastic opportunities for epilepsy research, and particularly rare epilepsies. And I hope we're here to help and support you, hopefully.
Thanks very much, Peter. That was really, really interesting. And great that you could summarize all of that very technical and scientific information in such a short time for all of us. Can I sort of ask one sort of quick question? And this sort of cuts across to Charlie's area as well. So in my Ring 20 hat, I represent a particular rare epilepsy that's associated with a ring chromosome. So ring chromosomes, structural variation of the genome. And there are many epilepsies, as we have Artie here as well as from Unique, many chromosome disorders where epilepsy is a key feature. And with these structural variants and these large CMVs, there appears to be lesser opportunity for research than in the SMVs. You know, the single gene mutations we're seeing, such as Dravet, the ASOs being applied CDKL5, etc. But with the more complex biology around these larger copy number variants, where we don't know the gene or the genes specifically, or the gene expression that's being altered, I guess what I'm asking is, in that wider sense, not just the Ring 20, but all the structural and these chromosome disorders associated with epilepsy, what kind of opportunity might there be with the sort of work you're doing in NATA? You did mention something about pathways and things, so that gave me some kind of glimmer of hope. You know, what kind of opportunity might there be there?
Yeah, thanks, Alison. Yeah, I agree. It's a tricky area because it's been limited, I think, by the models that are available. I think a lot of it's been driven by rodent models, which isn't always the best thing. I fully agree. And I think things like Down syndrome and things, which obviously, you know, large chromosome rearrangements, a huge amount of work has been put into making models that have the similar rearrangements. So I think these rarer CMVs and large deletions, it's quite a big enterprise to make a model because it's just for one single example. So I guess where we could offer is if this system's very good at doing combinations of knocking down different genes in different combinations. So I guess where there might be interest is saying, well, we don't know, this is a region that's deleted, so we don't know which genes involved. So the key thing is what's the combination of ones that are missing, which leads to a phenotype that causes disease, if that makes sense. So I think, and that's the golden bullet, is finding out which ones of those ones that are missing is causing the problem. So I guess the type of things we could help with is perhaps combinations of influencing genes along that in that region in different combinations. So you start to see which ones are leading to a phenotype which might lead to potentially seizures downstream, etc. That's one type of thing. So that's the kind of thing which genetically in a mouse something would take years and years and years, but you can do it much more quickly in a cellular model and do it that way. So combinations of gene deletions, for example, that's the kind of thing. I think structurally, looking at chromosome structure, physical structure is much more complex for sure. It's not quite what we can do at the moment. So that's sort of beyond. But I think certainly more of a technical aspect of interactions between genes would be feasible for sure.
Go on, Charlie.
Sophie's next.
Thank you, John.
Sorry.
Hi, Peter. It's a two-part question. Part one, are you doing anything on the calcium channels? Part two, anything on CACNA1C in particular?
Yes, so the field in general is doing both of those things. So we're not doing that ourselves. Yes, yes is the answer, I think, because they're obvious targets. And because we can do gain and loss of function type targeting, then there's room for directionality there. So yeah, so the answer is yes. We're in contact with people doing those kinds of things. There's, as you mentioned, as I'm sure Jonathan, who's in SYNGAP, will know, there's some of these things, there's quite a few different pharma companies going after the same target. So what we're here to do is not to compete, and is really to make sure we're facilitating and sort of, I guess, speeding up the work that people are doing. So we're in contact with quite a few of the companies which are targeting these and say, well, can we help? Can we integrate the data? If you're struggling, can we help you move forward? So the answer is yes, and yeah.
Can I drop you a line?
Yeah, of course, yeah, definitely. Yeah.
Thank you.
Yeah, Pete, that was really interesting. And hopefully Genomics England and NASA will be working together. That's the intention. I'm just curious, thinking about what Alison said. So Alison, forgive me if I get this completely wrong, but with the ring 20 chromosome, where the telomeres have attached to make the chromosome, there seems to be some idea that perhaps some of the genes around where that, outside of the telomeres, but around that area, are involved with epilepsy. I think we even looked at some in Ensemble.
Yeah, so CHR and A4 and KCNQ2, I think,
yes,
been implicated on the end of the chromosome. Yeah.
So what I was wondering is, how many genes can you look at in one go that might be, if you don't know which part of the chromosome specifically is involved, are you able to sort of knock down or knock up? Well, sorry, you know what I mean. Able to investigate those genes in that area to get an idea of which genes might be involved and might not be.
Yeah, definitely. I think that's the advantage of these new technologies, and also CRISPR, which you can do knockouts quickly. Is you can do combinations. So you could do a null hypothesis driven experiment and just do all the combinations. That's the kind of thing that's the scale which is possible now, and see where the interactions occur. And if your readout's good for a phenotype that's something that's relevant, then yeah, you can just do all the combinations. Definitely, you can do multiple things at once. Yeah.
So how many genes could you do in one go? Hypothetically, just I don't know.
It's not limited. I mean, the idea being is that each one's finding its own partner, as you know, Charlie. Each one's finding its own partner in the cells. So there shouldn't be competition. And also, you design them; they wouldn't get in the way of each other, of course, genetically or physically. So there's in theory there's no limit. With CRISPR, as you know, they've done dozens at a time, hundreds in fact, and that's the way they've made. I'm sure there's plans to bring back the dodo, etc. That'll be dozens of CRISPRs at the same time to change lots of genes at once. So yeah, so it's the same idea. There's really no limit, which is fantastic. It's hard to do. You need a big setup to do it, but actually these things are now routine tools, which in our time would be take months and months to produce, but now you can even buy libraries of combinations. So
thank you.
Charlie, can I just pose something to you again on the structural variants? So Genomics England 100,000 Genomes project, as I understand it, primarily or initially was mostly focused on using short read sequencing for whole genome sequencing, which, as we know, won't pick up a structural change. I was speaking yesterday from a guy from Oxford Nanopore following the Festival of Genomics, and they are telling me that they are working with Genomics England, and the nanopore technology that they apply can see these structural changes, which in long read sequencing that can't be seen in short read. And I think, you know, coming towards Peter's work and that NATA are doing, you know, if we could understand more about some of these structural changes at a genomic level, maybe we'd be able to give the guys, you know, at NATA and similar, you know, more clues to sort of what to aim at in their technologies. I just wondered what kind of opportunity there is for long read sequencing. Also, I spoke to BioNano, another company who are using OGM, and they can see ring chromosomes because they can see that the bits of the genome are in the wrong place, and therefore it infers you have a ring and other structural changes. So I'm just wondering what opportunity there is in Genomics England to look for more structural variations, and maybe to give us a bit more information, A, for therapeutic discovery, but B, or maybe the other way around, you know, diagnostic opportunity.
Yes, as I understand it, there is or will be some long read sequencing happening at Genomics England. I'm not entirely sure when or for whom, but certainly that is something that's being looked at. I do wonder whether perhaps one of the problems may possibly be that while you can sequence the structural variant, I'm thinking ring chromosomes, I'm not sure how that might be visible to anyone investigating it just yet. So in your browser, it might be difficult to actually display that information. I don't know, but thinking about it in the past, it's generally when you tend to look at a patient genome, it's a linear illustration of the chromosome that variant is found, and therefore a ring can't necessarily be displayed. So I don't know whether it might be sort of like a software problem that you can't do, you can't view it rather than actually being able to do it. But I'd be interested to know more about the technology that can identify the ring chromosome or how they do it. I don't know whether they're working with Genomics England or not, but that would certainly seem to be an important thing as, you know, as we get away from things like karyotyping. Hopefully they won't ever go out of fashion completely, because you know, in such cases, this is, you know, currently really the only way we can see these sorts of things. So it would be useful to know which techniques or technologies, work.
I'm really conscious of time. We've got 2 minutes left, and we just had a couple of extra points. Is everybody okay to go over by about 5 or 10 minutes, just so that we close off? Obviously, if Charlie and Peter have to leave, then I completely understand that. I'd just like to cover up any points. And was there anything burning that anybody particularly wanted to ask Charlie or Peter right now?
Just to say that I don't intend this to be a one-off. I intend to come to all of the rest of the meetings when permissible. So consider me now as a member of the group rather than an invited speaker. I don't know whether Pete's the same, or you probably got, I don't know.
No, definitely happy to come again. There's so many opportunities just to kind of just to hear, you know, concerns and ideas, and yeah, and him, you know, I can be fairly neutral here about, you know, what's happening in the field generally about sort of therapeutics, and be realistic about things. So that's, you know, if that's useful, then very happy to be involved.
That's fantastic from both of you. And would you both be open to, if anybody on the call here, or when we circulate the recording to the members of UKRET, if anybody has any questions for either of you, would you be happy for them to contact you directly?
Yeah, absolutely. Yeah,
that's fine. Yeah. Yes, I just reach out. I'm not a clinician, but so yeah, it's general queries around the sciences is fine.
Yes, I should also mention I'm not a clinician, but yeah, very happy to answer questions where I can, and if I can't, I'll try and find someone who can.
Yeah, I don't see pictures of your rashes, put that way.
That's incredibly helpful, and you know, I just want to say thank you both very much for coming along today and putting, I think, some really interesting points to this group, and giving us lots to think about. And I don't think anybody would be adverse to you coming back again and continuing the conversation and keeping us updated and seeing how we can work with you, with both of you, on those. So I just want to finish off then today, coming back to a couple of updates on some points that have been ongoing. I know we still have Daniel with us. So maybe can I pass to Daniel? Do we have any updates on the Conditions Insights report work with the DWP that we were working on the end of last year? Hi, Daniel. I don't know if you can hear us just at the moment.
Sorry, yeah, I can hear you. Can you hear me?
Yes, we can now, thanks.
Oh, right. Okay, sorry. So yeah, I haven't had anything back from Chris at Athos yet about the Condition Insight report, but I am due to meet him in the next couple of weeks just for a general update anyway. And ahead of that, I've asked for an update on the Condition Insight report for rare epilepsies. So I'm hoping to have something to share with the group very, very soon.
That'd be great. It's good to know that you'll be following up. We'll be really interested to see, next steps on that. So thank you for that. I know Rosemary wasn't able to join us today, but I know and I hope lots of us are looking forward to the event at Young Epilepsy next month, now March, and lots of us will be exhibiting, and be a great opportunity to meet with everybody there. I think if anybody hasn't already shared on social media, I do know that when we were at BPNA the other week, that Young Epilepsy are still trying to strongly encourage more delegates to attend, more clinicians or epilepsy nurses or medical professionals in the field to actually attend the event. So sign-up's been a little bit slow. So anything that we can do through the communities we work within that you can circulate, promote for those medical professionals to attend, because for it to be a great event, we do need a good attendance. So please feel free to share. I've actually having it shared through the Epilepsy Nurse Specialist Network newsletters, and I know I've seen some of you have been sharing on social media. So looking forward to that. And then very lastly, I know that Jenny and Stephanie weren't able to join today, but just wanted to give you all a very brief update or debrief from our BPNA shared exhibition stand, in-person stand, the other week. There were the three of us that managed the stand, albeit I was only there for one day, and Stephanie and Jenny did most of the work over the 3 days. So huge thanks to them. I know some of you were also in attendance with your own stand, so it was great to catch up with some of you. We did have, particularly on the day that I was there, some key attendees that came up to us on the stand, and that was on the Thursday when there was the hour and a half that was dedicated within the programme to come and talk to the charities. I think generally the charities that attended, the layout of the event, we were upstairs in one of the meeting rooms, and there wasn't a huge, I think generally everybody sort of said there wasn't a huge amount of passing traffic, and that the attendees tended to circulate more downstairs where the main larger organisations were, that paying organisations for their stands were placed. So I think some feedback to BPNA on that, because the whole opportunity to exhibit needs to be an opportunity to network, and if people aren't going to come and see you at all, and not even going to visit the room, then that's really lost. And I think those of us that had a virtual stand, which of course was free. I mean, I know from a Ring 20 perspective, we didn't have one visitor to our virtual exhibition stand, and that's not unusual, but I don't know if others had better attendance. I'd really love to, I think the group would love to know, because again, you know, we should feed back to BPNA. You know, it can be a lot of work to do these things. It costs a lot of money to attend in person. And if you're not going to get the payback from that networking opportunity, then, you know, we have to be really careful as charities. We did put up, as you can see in the little picture of us in the corner, we did have the banner made, and all of those of you that shared your logos are all on that banner. I have paid for that out of Ring 20 funds, but we're by no means anywhere near the largest organisation. In fact, we're probably one of the smallest organisations in this UK Rare Epilepsies group. It cost, you know, almost £50, and it would be really lovely if, you know, a small contribution, the cost of a cup of coffee, was offered from those of you to share the cost of that. And those of you that are happy to contribute, you know, I can share some PayPal details just to make that a little bit easier, because it's a shared initiative. And, you know, the three of us that were on the stand were talking UKRET, not about the specific charities and diseases we represented. And then sort of finally, just thinking about, you know, if we do a stand again in the future, or we represent as this network of UK Rare Epilepsies together, you know, should we consider any other documentation, maybe a small leaflet or a couple of different leaflets, you know, something for patient groups that we haven't yet got to join us, you know, what we are, what we do, what we're about, how they can sort of become part of us. And also, you know, for medical professionals or pharma companies or other organizations that we want to talk to, engage with and collaborate with in the future, you know, again, what we're about, but how they could come along to meetings like this and talk to us, share their work, their opportunities, and how we could work together. So, you know, and any other ideas that anybody has. So I'm really conscious of time. If anybody has any immediate thoughts, you know, happy to take those now. But other than that, you know, please share back to the group, you know, any thoughts or opportunities for how we can make the best of this network that we're starting to build, and go from strength to strength, really. So any final thoughts before we close off on that?
I'd just like to say thank you for that, Alison. As you know, my vice chair couldn't make it because she was ill, which is a shame, as I couldn't. So, and I don't know, did I send you a logo to put on your thing, on the banner?
I believe Bechet's, you're
right,
on
okay,
there.
right.
Yeah,
Well,
yeah.
I'll send some money. Yeah, I apologise for that. But I agree with you. A sort of little leaflet, maybe showing the all the different conditions we're all ganging together on might be a good thing for future shows. And I mean, I don't know whether I've one of my part-time admin ladies who's pretty good at putting things together. If you need any assistance there, she could probably help out. So, but you've probably got your own access to them. But thank you for doing that, because it was really useful.
Thanks, Tony. You know, appreciate any help. You know, although I'm kind of coordinating this little network, you know, it really, you know, I could use the help and contributions of others here. So anybody that's happy to help with any of these initiatives, you know, either with the design or creation of you know, just some small assets. And also, you know, we probably need to start to think about moving towards some kind of joint funding application, just to be able to get a little bit of money, formalize this network, get a little bit of money to put towards, you know, just basic things that we need. Just to say that I have put in for the London Met University Social Enterprise Clinic to see whether they would take on building us a very simple website. So I'm waiting to hear back. That's been put to the students, as I understand. I think, in the last week. So I'll wait and see, and I will update everybody if that's successful. Create us something really, really simple to get us started and get us out there, which would be fab. Anything from anyone else? So just very, very finally for a sign off. So this was something that's just come in very, very recently, but I put it on the quick point on the agenda. So I've been involved. I happen to know the people from Aspire Biosciences, who are a fairly new biotech. They organised an event last year that I attended representing Ring 20, and it's a networking event that puts 10 patient organisations in contact with something like up to 10 partner organisations, pharma industry, to sort of, we get an opportunity to pitch what we want and need, what our unmet needs are, and to see whether these partner organisations have anything to offer us and could collaborate with us into the future. They're organising a similar event again for neurological disorders, which obviously includes the epilepsies, and they're really keen to have us back, but really interested, and some of the potential partner organisations that they are trying to bring into this event are also interested in more of a collaborative epilepsy opportunity. And the way that we pitched actually for Ring 20 last time was not just for Ring 20 on its own, because we're really, really ultra rare, and we know that we're not super exciting on our own, perhaps to some of the researchers and academics out there, but as a wider cohort of, say, either ring chromosomes or, as I've mentioned earlier on the call today, structural variants associated with epilepsy, so chromosomal disorders associated with epilepsy, which is really an untapped field of discovery. I wonder whether anybody else would be interested in partnering and in a sort of a joint application into this event, joint opportunity to participate. I can certainly share more details. We have to pitch with a little video pitch by later this month, and then we have to put some info together. So just kind of putting that out there, but if anybody's kind of interested, you know, really happy to share that opportunity with others, because I think, you know, collectively, potentially, we might have more success.
Sorry, thanks, Alison. Artie from Unique speaking here. I think you did email me and Sarah yesterday about it, so I think we were just going to have a chat about it today. But if you've got a few minutes after the call, if I could just have a few minutes just to learn a bit more about it, and then I can take it back to Sarah if that's okay.
Yeah, certainly. We'll stay on the call and we'll just catch up. Thanks, Artie. Okay, so I think if there's nothing else, we are sort of 15 minutes almost over, but, you know, a really useful, really interesting call. Thank you everyone for joining. Thank you all for staying, and you know how to get in touch, and let's keep this network going and keep the conversation going.
Thanks, Alison. Bye,
Thanks
everyone.
very much, Alison.
And thank you again,
Alison.
Charlie
Thank you.
and Peter.
Thank
Bye.
you. Bye.
Yeah, thanks, Charlie, Peter. Awesome.